In this video, we will discuss about how this integrin alpha V involved in C1GalT1 mediated invasion in pancreatic cancer cells. So far, we have understood in the till our previous videos that C1GalT1 knockdown affect the glycosylation profile on alpha V, alpha 5, and beta 1 domain of the integrins. So, in this video, we will understand how this alpha V integrins out of these three domains that we have discussed in the previous videos plays an important role towards C1GalT1 mediated invasions in pancreatic cancer cells. So first what they did is they use the cell lines, that is MIA PAC1 for overexpression of C1GalT1 and HPAF2 and HPAC for this temporary knockdown of C1GalT1 using this mRNA, C1GalT1 mRNA. And then they check the expression level of three domains, that is beta 1 alpha 5 alpha V and alpha 5 beta 1. And what they are able to find out is that both in the case of overexpressions and the suppression of C1GalT1, they do not see any significant changes in those domain. Indicates that it's not that the overexpression of this domain cause the change in the glycosylation profiles. Thereafter, they wanted to check the invasions properties of these cells when one of this domain has been knocked down. So what they are able to find out is that in the case of HPAF2, when the alpha V is been neutralized using anti-alpha V antibodies, they see a significant decrease in the invasions for both this HPAF cells and HPAC cells compared to the control, that is IgG. However, when they knock down alpha 5 using anti-alpha 5, they are able to see significant changes in HPAC, but not in HPAF2 and MAC-PACA2, MIA-PACA2. And thereafter, they check the integrins signaling pathway phosphorylations, that is FAK molecules, integrin signaling pathways FAK molecule phosphorylations, that is FAKPY397. And what they are able to see is that they see a slight change in the FAK phosphorylations when they use anti-alpha V, but this change is not significant. But when they did when they inhibit this anti-alpha V using some inhibitors, they are able to see significant changes in the FAK, and those results are significant changes in the FAK phosphorylations, and those results are shown in the supplemental figures. And thereafter, what they did is they check the glycosylation profiles using pull down assay with the help of this VVA lectin. And what they are able to see is that in the case of alpha V, there is a significant increase in the VVA signals or this TN antigens in the C1GalT1 knockdown for both HPAF2 and HPAC cell lines. And these results are their consistent, and this increase in the signals is also statistically significant compared to the alpha these VVA signals in the alpha 5 integrins. So this substantiate their finding that alpha V plays an important role to C1GalT1 mediated invasions, and when C1GalT1 has been knocked down, they see an increase in the O glycosylation profiles or increase in the TN glycans on this alpha V, and that cause their reduced C1GalT1 mediated invasions in the pancreatic cancer cells. Or that's why we see that in the C1GalT1 knockdown cells there is a reduced migrations and invasions that we have seen in the panel C and the previous videos. So what they finally conclude is that how this C1GalT1 helps in the migrations and metastasis is that this alpha V domain of the integrins, alpha V integrins will have this O glycan profiles, and that interact with the ECM and thereby triggering this FAK phosphorylations pathway, and that will cause tumor growth and metastasis. And when the C1GalT1 has been knocked down, there is a change in the O-glycosylation profiles, that is more truncated TN glycans, more truncated O glycans are expressed, and that prevents its interactions with the ECM, and thereby these signaling pathways cannot be triggered, and that leads to the decrease in the tumor growth and metastasis. So this is their final outcome from their studies. So I hope you understood how this C1GalT1, how this integrin alpha V plays a role towards C1GalT1 mediated invasions in pancreatic cancer cells. If you need to understand more about this paper in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.